Class. Today, this lesson is going to be about G-coupled protein receptors, and this is for the signal transduction presentation. My name is Josiah Weimer. This is Chem 530 Biochemistry, and here we go. So as an introduction, it's important to highlight that one of the most important parts of biochemistry is cell signaling. And basically, simply put, cell signaling is the transmission of messages from outside of the cell to inside of it. And this cell signaling can take place between cells, or it can take place entirely intracellularly. Cell signaling performs countless functions in the body. This can include protection, similar to what you would see on white blood cells, signaling for antibodies to take out harmful antigens, for regeneration, so you see a lot of damaged cells, and those cell signals can influence the body to create more cells, and then also communication. So that you see that in a lot of tissues. You see it when there are antigens in the body. There are just multiple examples. One of the most important cell receptors found within the cell are the G protein coupled receptors. And these are the most common second messenger systems in the body. So a second messenger system is basically whenever a ligand or a molecule binds here to this receptor, and then that signals another chemical in the body to produce a certain effect. And that's what we call a second messenger. And at the most basic level, the G protein coupled receptors will receive the ligand and initiate the effector. And that effector is what's going to kick off the second messenger system. So in my presentation, I'm going to outline the following points: the function and mechanisms of G protein coupled receptors, how these G protein coupled receptors will manifest in the body and how they contribute to disease, and then finally the applications of the signaling system in medicine along with future research. So one of the most important parts of learning about GPCRs is learning about the discovery of them. And it's important to note here that for a while we didn't really know a lot about GPCRs because they were really hard to isolate. And this was due to their structure, which I'm going to go into in a second. But you can actually kind of see it here in this picture. It's just a bunch of alpha helices. And those alpha helices are stable on their own, but the way that they're configured within the G protein receptor, they're just kind of loosely put together. So it was very difficult to isolate them because outside of the cell membrane that they were put in, they would just kind of fall apart. And much of the important discovery of GCPRs came through the study of what are known as radioligands. And what radioligands are basically, they're like ligands. They'll bind to the receptor, kind of like what you'd see in here. But they also had a radioactive tag on them, making it easier for researchers to identify where these receptors are in the body and what the nature of them are like. And one of the most important people in the G protein discovery was Dr. Robert J. Lefkowitz. And he made a lot of his discoveries in the 70s and 80s. But a lot of his work revolved around cloning and sequencing beta-adrenergic receptors, which belong to the family of one of the most basic forms of G protein receptors. And he was able to identify certain common structures in GPCRs and distinguish them. And another important advancement in technology with GPCRs is X-ray crystallography. And that's kind of it's a way to visualize the protein in 3D through the use of X-rays. And it's allowed us to visualize these receptors much more effectively. All right, and that brings us to the structure of GPCRs. And the structure, similar to other protein receptors, the structure of GPCRs are one of the most defining characteristics because the structure really does define the function of the protein. So one of the more notable things on a GPCR are the extracellular N-termini. And you can see that up here, actually right there. And that part's just kind of going to stick out of the cell. And it's important for a couple of reasons. It's responsible for extracellular signaling within the body. So that means that all the interactions with other cells, the N-terminus is very often involved with that. And it also functions as a ligand receptor, and that's a place that has a very high affinity for certain chemicals, also known as ligands, which will bind to that receptor and then start this chain reaction of events that lead to the second messenger response. And then another of the really important ones, actually the namesake of the GPCR, it's the 7 transmembrane alpha helices. Often GPCRs are also called 7 transmembrane proteins, and it's because of this. They have 7 alpha helices that span throughout the cellular membrane. And these places can also help with the ligand binding, but they also do communicate with each other as well. And it's the most variable region in the GPCR, and it's actually one of the most effective ways to distinguish the families. And then that brings us to the intracellular C-terminus, and you can see that down here. It's just this little tail at the end of the G protein receptor, and it's going to actually be responsible for activating the G protein, and we're going to get into that a little bit later here in this presentation. All right, and then another important part of the GPCR structure is also the G protein. And G proteins are named after the chemical that they bind to. It's a nucleotide known as guanine, and whenever it's bound with a sugar, it is known as a guanosine. So it's a guanosine protein that activates and propagates a signal. And you can kind of compare it to a light switch that way, because whenever it has a GTP, or guanosine triphosphate, bound to it, it's actually going to turn on like a light switch and activate. And there are two main types of G proteins. We have the heterotrimeric G proteins, and then we also have monomeric GTPases. And we're going to start with the heterotrimeric G proteins. This is the majority of G proteins you're going to find in the body. And those G proteins, they come in 3 parts. Trimer means 3. And they have an alpha unit, a beta unit, and a gamma subunit. And you can see them right here. You got your G protein coupled receptor with all your alpha helices, and then you have these 3 subunits. And whenever they're together, there's actually going to be a region on the alpha subunit of the G protein that is going to be very receptive to GTP. And you can see that right here. It doesn't have much of an affinity at all for GDP, so it's going to kick that right off. But you can see here that GTP will hop straight on, and whenever that happens, the light switch happens, and the alpha unit of the G protein will be activated, and it's actually going to detach. And we're going to get more into this mechanism as the presentation goes on as well. But the important thing to remember at the moment is that the heterotrimeric G proteins have these subunits that interact with each other based on what the ligand is doing on the G protein receptor. Then we also have monomeric G proteins, and those consist of only one subunit, so you're not going to see this division as you would in a regular G protein. But essentially, the activated protein will help act as a second messenger and interact with other factors downstream, which allows for more cell signaling. And it also functions as a transport for chemical receptors, or messengers, I should say. But interestingly, one One of the earlier forms of G proteins that were discovered were actually known as RAS, and they were found in this rat, and they're actually discovered because it was a mutated form of a G protein, but it contributed a lot to cancer growth. Just an interesting fact. And the important takeaway here is both types of G proteins are going to relay messages to the intracellular targets. That puts us at a very important section, very interesting section, and that's the function and the mechanism of GPCRs. Because structures tell you a lot about the cell, but the functions are really what's going to drive it home, especially if you're teaching a class. So, one of the first steps to the GPCR or the receptor binding is going to be the ligand binding to the receptor, and that ligand is just a chemical messenger that is specific to this GPCR. And what it's going to do is it's going to either bind to these alpha helices or to the N terminus, depending on the member of family that the GPCR is from. And that's going to activate the GPCR. And whenever you see a GPCR activate, there's going to be something that happens known as a conformational change. And basically, that just means that the GPCR is going to kind of change shape. It's going to extend that tail, and you can kind of see that here. It's going to extend the tail, and that tail is going to bind to the alpha subunit of the G protein. And this leads to the G protein activation, kind of that light switch that we were talking about. And you can see kind of right here that up until now, this G protein is not activated quite yet. But whenever it is activated, it's going to have GTP bind to it. It's going to kick GDP off. And then what's going to happen is the alpha subunit, whenever that alpha subunit of the G protein is activated, it's not going to want to hang around with the gamma and beta units. So whenever it has that energy molecule on it, it's going to dissociate because its affinity with the energy molecule is greater than its affinity with the subunits. And then what's going to happen at the most active phase is that the G protein is going to hydrolyze the GTP. And that what that means is a water molecule is going to come in and it's going to break one of the phosphate groups off. And that's going to cause an inorganic phosphate to be resulted along with the GDP. But most importantly, it's going to be a release of energy. And once that energy is released, the alpha unit is going to develop that affinity again for the gamma and beta subunits. It's going to bind right back on, and that's going to perpetuate the cycle. Okay, and that brings us to the G protein. And we're just going to look at this in a little more depth. But the subunits of the G protein, once it's activated, it's going to bind to an effector such as adenyl cyclase. Now, I realize that there are other mechanisms that allow a G protein to bind, but that's one of the more well-known ones, so I'm going to use that as an example. But you see the ligand binding here? It's going to have, it has the GDP, kicks that off. It dissociates whenever the GTP binds to it. And then what the alpha unit is going to do, it's going to float down to this adenyl cyclase right here. And that adenyl cyclase is going to be the next step in the chain reaction that is going to allow for the message to be sent to the other part of the cell. And And at the end of this cycle, the G protein is just going to deactivate, and that's after the energy is released. It's going to reunite with the beta and gamma subunits, kind of like what I was talking about. Another important thing to note is that G proteins and G protein coupled receptors are not the only ones that interact with each other. There's another important family of proteins known as beta arrestins. And similar to what their name sounds like, they're going to inhibit the GPCR or the G protein coupled receptor from reacting with the G protein. And that's going to reduce levels of second messengers. And it's important to remember that the GPCRs are very important in a lot of regulations. And with cell signaling, it really goes both ways. You can have the signaling kick up certain functions such as protein production or the breakdown of glycogen into glucose. But you can also have the opposite, where those processes are inhibited and the reverse will happen, so glycogen will be built up, or those proteins will stop being made because the body no longer needs them. It's all part of negative reinforcement. All right, and this brings us to the signaling pathways of GPCRs. These GPCRs act in a second messenger kind of system. And what they do is they're the first step in this cascade, which is designed to amplify signals that are received from the outside of the cells. And there are really two main ways that the GPCRs transduce signals, and we're going to talk about them right here. The first one is known as the cyclic AMP pathway, or CAMP. We can call it CAMP. But what happens is that activated alpha subunit of the G protein is going to bind to adenyl cyclase. You can see that right here. It binds to the GTP, dissociates, jumps over here, binds to the adenyl cyclase. And that adenyl cyclase is just a machine, it's an enzyme that's going to take ATP from the cell and convert it into cyclic adenosine monophosphate. And that's going to be a second messenger. And what that second messenger is going to do is it's going to influence other proteins such as pKa or protein kinase A. It's going to influence it to produce more of a signal, or it's going to tell the protein kinase A to start creating certain proteins. And this protein kinase is responsible for phosphorylating proteins. So it's going to change their configurations so that they're available for the body to use. And something important to note about pKa is that it acts only in the presence of CAMP. So if you have this inhibiting system, what's going to happen with the pKa is it's not going to be acting if that inhibiting system is binding to the GPCR and stopping CAMP from being produced. And here's a clinical example. We have epinephrine, which is going to bind to a beta adrenergic receptor. Epinephrine is our ligand, and that beta adrenergic receptor is our GPCR. It's going to activate the G protein, and that G protein will bind to adenyl cyclase, start producing CAMP. That's our chemical messenger, and that's going to activate the pKa to make proteins that our body needs, such as glycolysis, enzymes that take part of glycolysis. And that leads to a sympathetic cascade. You're going to see the blood pressure go up because of smooth muscles contracting, and you're going to see reduced blood flow to the digestive system, and you're going to see more in the skeletal muscles. Okay, another important signaling pathway of GPCRs is the phosphatidyllinositol, or I'm going to call it the PI4-5P2 signal pathway. It's a lot easier. And then that PI4-5P2 is a fatty chemical compound found within the plasma membrane of the cell. So basically what this pathway is going to do is you're going to have your ligand bind to the receptor, and we have a G protein, and this G protein is a little bit different than the one found in the cyclic AMP system or CAMP system. And this one is known as GQ, but it has a very similar function. And what it's going to do is instead of activating protein kinase A, it's going to activate an enzyme known as phospholipase. And the function of that you can kind of see in the name. And what it's going to do is it's going to hydrolyze or break down that fat molecule with a water molecule. And it's going to make two different compounds. First, it's going to make inositol 1,4,5 triphosphate, we're going to call that IP3, and diacylglyceride, DAG. And these two are very important to the cascade, and they have distinctive functions. We have the IP that binds to the endoplasmic reticulum, releasing the calcium ions into cytosol, into the cell. And then we have the DAG and calcium ions working in conjunction to activate the protein kinase C. And that's just going to phosphorylate or activate multiple proteins within the cell. And one of the most important places you can see that are in smooth muscle contractions in the GI tract. So if you need peristalsis, which is the way that the muscles push food along the system and then churn it inside the stomach, what you're going to see is you're going to see a ligand bind to that receptor right there, and then you're going to see the G protein bind with GTP, and then attached to this phospholipase C, it's going to take molecules from in here, and what it's going to do is going to produce DAG, and that DAG is going to work with the calcium. It's going to stimulate this protein kinase. Okay, and then another important part of signal transduction is knowing what it does in the body. So we already looked at it at a biochemical level, but we're going to kind of zoom out at a bird's eye view here. And it's important to note that there are many GPCRs within the human body. We have over 800 types found within the human genome. And to put that in perspective, GPCRs account for 2% of the human genome. So these receptors really help us function in our day-to-day lives. So it acts as receptors for a number of ligands. So you have light particles, and that's typically seen in the optic senses. A lot of these are sensory. We have olfactory, which has to do with your smell. And you can see why second messengers would be really important in here, because if you're receiving information from your environment, you're going to need your body to amplify those signals and then produce action based on them. So if you see a baseball coming at your head, you have the light receptors, and those are going to bind to your GPCRs, and that's going to help your brain compute there's a ball coming at your head. And that's going to actually going to have adrenaline sent to your beta adrenergic receptors. And those adrenergic receptors are going to cause that cascade that will make you duck really fast so you don't get hit by the ball. And then we have neurotransmitters. And one of the most important things about the GPCRs with neurotransmitters is the release of calcium ions, because those are important in helping regulate how much of the neurotransmitter is being sent to the postsynaptic cleft. And then we have hormones, and those are just chemical messengers that are dumped straight into the bloodstream, and they're responsible for keeping our body in a homeostatic balance. So it can be seen that GPCRs are very important, and they're going to activate second messengers systems, and those will initiate cascades all over the body. Those can include secretions from cells, ion channels opening, or even DNA transcriptions, which are all necessary for life. Okay, GPCRs are one of the main ways that we classify them are based on structure. And the most common family of GPCRs are Class A, also known as rhopsidins. And it's just got the basic look of the GPCR. So here's an example right here. You see the N-terminus up here. You see all our alpha helices that are bound together, and kind of sandwiched in here we have our ligand binding site. And then we have our C-terminus, which is responsible for activating the G protein. And one of the biggest functions of these rhopsidins are sensory, such as sight and smell. But they're also important with hormones and neurotransmitters. And And you'll see a lot of hormones in other GPCRs, but these are also involved because they're just the most common. And this means that they're the greatest therapeutic targets of medicines because you got to remember these GPCRs are very important to um, all sorts of functions and across the body and pathways. And two examples of the pathways that rhopsidins undergo are the CAMP pathway and the PI4 5P2 pathways. Then we have our class B, and these are known as the secretin GPCRs. Those are around 60 members, so it's a substantially smaller group. But these are going to have a larger N-terminus than the classic GPCRs. And this is important because these GPCRs are involved in hormonal homeostasis. So they're going to need to have ideal communication between them and other cells because hormones are all about communication. They're chemical messengers. And these are involved in cell adhesion as well. So they do help with the cells having a certain structure in our body. And some of the most important ligands that bind to these GPCRs are glucagon, secretin, and calcitonin, along with growth hormones. Okay, that leads us to our next group, which are known as the metabotropic glutamate receptors. And these receptors are going to regulate cell excitability in the CNS. The CNS in the brain, spinal cord, all that, that's really where you're going to see most of these. That's where you're going to see most of these receptors. You're going to find them in the presynaptic and postsynaptic membrane. And they have a larger N-terminus, and this allows for glutamate binding. And glutamate is an excitatory neurotransmitter, and it's responsible for how we think and the transmission of information in our mind. So it's very important. And in order to regulate that, it's kind of on a tightrope between inhibiting and allowing it. So these are very important. And some of the places you're going to see it are GABA receptors, which help with the regulation of pain, and then also calcium receptors, kind of like what we were talking about with the synaptic cleft. It kind of helps regulate the neurotransmitters. And you can see right here that it's much bigger, and it kind of looks like a Venus flytrap. And you got that glutamate molecule in there. And then we have a smaller receptor family, and this is known as the frizzled receptor family. And this one we didn't know as much about until recently. But this one is responsible for receiving a ligand known as WNT, and this affects a messenger known as beta-catenin. And researchers have found that beta-catenin is especially important in embryos, and it's going to help especially develop their trunk, and it's going to help the cells become what they need to be for that baby to grow. But another important thing to note is that in adults, these frizzled receptors can still be active, but often they are somewhat harmful. And they're often seen in cancerous tumors such as breast cancer and prostate cancer, which are genetically linked. So if you're genetically predisposed to having those kinds of mutations, it's definitely important to be checked on that. All right. And while GPCRs have a lot of importance in bodily functions, they are responsible, their malfunctions can be associated with a lot of diseases. And you're going to see this more at a biochemical level, because a lot of times whenever the GPCRs are malfunctioning, it's because of a mutation found within them. And there are typically two kinds of effects that these mutations can have. It's either loss of function or gain in function. So if a protein structure is defective, it means that it just won't work at all. So there's no regulation and no initiation of anything. So it's just not going to function the way it should. And one example of that is color blindness. And this is caused by defective GPCRs that are found within cone cells. And then we also have thyroid adenomas. And those are an exact opposite problem from what we saw in the loss of function. This is an abnormal gain in function, and this can take place whenever there's a point mutation that creates a different kind of amino acid instead of just terminating it early like you'd see in a nonsense mutation. And you're going to see that in an overactive GPCR in the pituitary. So if you're seeing too much thyroid stimulating hormone, that can be associated with symptoms that you'd see in people with hyperthyroidism, so anxiety, irritability, increased metabolism, even hair loss. And then another example is osteoporosis. Since the GPCRs are very important in bone remodeling, especially the class B ones that regulate the calcitonin, if those are mutated, it can allow that bone remodeling relationship with the osteoclasts and the osteoblasts regulating each other. It's not going to work the way it should, and those osteoclasts can become overactive, and that'll just eat away, causing the bone to be a lot more brittle. And the main takeaway from this is that by understanding the contribution that GPCRs have to their disease, researchers can understand how to target and treat these disorders. And on the opposite side of the coin, even though GPCRs can be the cause for disease, they can also be an agent for good because they're one of the most common targets for pharmaceutical drugs. They're ubiquitous in the body, which means that they're expressed everywhere. And it's been shown that over 30% of these pharmaceuticals target GPCRs. And medications can bind either orthosterically or allosterically to receptors. So an orthosteric would mean that this medication would bind directly to the active site of the GPCR. So that's just going to be what's called competitive inhibition. So if it's looking to inhibit a GPCR, it's going to bind where the ligand should typically be seen. And one example of that is the opioid. And those opioids have a high affinity the GPCR receptors have a high affinity for endorphins, and this allows the drug to bind to the receptor really quickly, and it prevents these G proteins from signaling pain responses. And then we also have allosteric inhibitors, and basically what those do, they don't competitively inhibit, but what they do is they change the thermodynamic function, and they cause the site to conform around it. And that's actually going to cause this site to not be able to take the ligand that it would normally be able to take. So those can be useful as well. Another example of medicines found with GPCRs are antihypertensives. So those will bring down blood pressure. And they're going to bind to beta adrenergic receptors. This is another orthosteric example because it's going to bind straight onto the place where the epinephrine would typically bind. And it's going to prevent this cyclic AMP messenger system from stimulating the sympathetic nervous system, kind of like what we talked back here about. So what would happen is this would be the beta blocker here, and it would bind straight here, so the epinephrine couldn't bind there. And that's going to prevent CAMP from being produced, which will prevent the cascade of sympathetic events from happening. And then we have a lot of other drugs with GPCRs, and that includes antipsychotics and antihistamines. And those are going to interact with GPCRs in such a way that it brings down those natural responses from the body. So this brings us to our conclusion. GPCRs are integral to virtually every process in the human body. And by understanding these receptors, we can also better understand how the body works. And with our God-given brains, we can also discover many other technological applications. And we have one example here. GPCRs, when we engineer them with green fluorescent proteins, what we can do is we can map neurological interactions that are taking place in the brain. That's been a difficult point for a lot of researchers because it's kind of hard to get into the brain with imaging at that molecular level and see what's going on between neurons. So if we tag it with this green fluorescent protein, this is going to help us actually have a better way of seeing what's going on in the brain, and that can lead to excuse me, that can lead to a treatment of many diseases, and then also highlighting, for instance, if someone has Alzheimer's, how their neurons are firing and where the location of certain beta amyloid plaques would be. And whenever we couple this with imaging techniques such as fibrophotometry or two-photon microscopy, we do have a very good potential for mapping these interactions. And this brings us to the end of the presentation. And the question I would like to leave you with is, how can we use our knowledge of the human body and its mechanisms to better serve mankind? That's all I have for today. Thank you for watching.